Thanks very much. Well, yes, so I'm from the Rosalind Institute, which is was the home of the most famous and the only famous sheep in the world, Dolly the sheep. And we work on animals, and specifically my interest is research in the chicken, but I'm funded by the BBSRC, the Biotechnology and Biological Sciences Research Council, and I'm on the council of that, of the BBSRC, and they support fundamental and strategic research in animal and crop breeding. And so I've developed a real interest in the application of new technologies, not only in animal breeding, but in crop breeding as well. And the BBSRC has as one of their main strategic pillars is agriculture and food security. So they're very interested in funding research which will bring new technologies much closer to success in plant and animal breeding. Okay, so today I'm going to talk about, I was asked to give a talk because I gave a presentation to the all-party parliamentary group of science and technology at Westminster in the summer, and I was asked to basically do the same talk, and the talk's about genome editing, and you probably come across that in the papers. Every week or two there's a new story about the new technologies. So I thought I would start off by explaining what the technology is, and then I'll give you some examples of how it's being applied, and I hope that you'll begin to see how we have new technologies that are coming along which will really provide additional tools to crop and animal breeding that we really must consider. Okay, so my first slide is a DNA molecule, and we know that DNA is wrapped up into chromosomes, and that this is very complex. Humans have about 1.8 meters of DNA in every cell. Wheat, the genome is 5 times longer. That each chromosome is a linear array of genes, and that's what we're dealing with when we're doing plant or animal breeding. And the new technology of genome editing is, the basics of it is that we now have a tool that will allow us to go into the genome of wheat, for example, or in my case chickens, and identify a single gene that we want to change. So genome editing involves a molecular tool, we call them molecular scissors, and they'll go into the chromosomes, identify a specific sequence, and cut the DNA at that sequence. And if you cut DNA, it's always repaired, and when the repair happens, it often forms a mutation. So it's a new way of introducing genetic variation into a plant or an animal. Not only can you make mutations, which usually, but not always, will stop a gene from functioning, you can make a very specific change. So if you want to change a gene from having one characteristic to another, you can make a very simple change of a few bases of the DNA using these genetic tools. And the most common tool that you'll read about, and we hear about it in terms of repairing human genetic disease, but it's exactly the same tool that is being used in plants and animals now. And they're called CRISPRs, and I'm not even going to tell you what that stands for because I keep forgetting what it means. But it's an enzyme that comes along and will find its way to a specific sequence in DNA, and it will cut it, and either cut it and it'll be repaired to give you a mutation, or it will cut it and you will make a very specific change. So CRISPRs are the most recently developed molecular scissors. They're extremely efficient at finding the target sequence and cutting DNA. And that cut DNA can be repaired to give you a novel mutation, or it may be repaired to give you a precise genetic change. So try and remember what I've told you about this technology, because I'm going to give you some examples. And it's because we have this technology that these examples I'm going to give you are now being developed. And there are two things about it. One is it's very, very specific, so we're making a genetic change that we design. And the other is it's very, very efficient, so it's very easy to make these changes. We just have to work out what changes do we want to make and how do we want to use them. So the first example is a work from a Chinese group that was published a couple of years ago, and it's about powdery mildew in wheat. And as we know, wheat has three genomes. So there's a gene called TAMLO, that if you knock it out, you stop the wheat from getting powdery mildew, but you have to knock it out in all 3 genomes that make up the wheat genome. And what they did was they used a CRISPR to knock out this gene in all the 3 genomes at once. And at the bottom, I hope you can see it, is wild-type wheat, normal wheat with a powdery mildew infection, and then their experimental wheat, where they've knocked this gene out and there is no infection. So it's possible that you could sequence wheat for lots of different varieties and find one that had that mutation in it. You would then have to cross it into your elite breeds to get the resistance to powdery mildew. With this tool, you could go in to an elite variety and introduce the mutation that makes them resistant to powdery mildew. And to just give you one example of an animal, because I work on animals, and this is where researchers had identified a protein that is required for the PRRS virus to infect pigs. So they went in with a CRISPR, they knocked the gene out, and they've shown that these pigs are now resistant to PRRS. And PRRS is a major pathogen of pigs in Europe and North America. And what's interesting about this was that this work was carried out at the University of Missouri, but with the animal breeding company, Genus PLC. So animal breeding companies are beginning to consider using the technology now. So this is an example that I got from the John Innes Center. And there they're working on the problem of pod shatter in oilseed rape. And at the moment, they've only got as far as modeling it in Arabidopsis, which is a model brassica. But they could easily transfer this technology to oilseed rape. And what they've done is compare wild type, and they know the gene that is involved in pod shatter to release seeds in Arabidopsis, and it's the same gene in brassicas. And they've mutated that gene, and this is a diagram that shows you the pod. And there's a particular mechanism that allows the seeds to be popped out. And in normal Arabidopsis, you can see that there's a space opening here, the seed will be popped out. In the mutant, they do not get that space forming, so the seed cannot be released. So by making a single mutation, you can change a plant from being one where the pods shatter and the seeds are released, to one where the seeds are retained. So the idea is that if you introduce this mutation into oilseed rape, you would stop the losses of seed from pod shatter. Another example, this time from Rothamsted research, is where it's a more complex. Yes, can I have some water? Thanks. A more complex of using the genome editing technology and what tends to be referred to as old GM. This is where they're going to introduce new characteristics into oilseed rape, well, into an oilseed crop. So I work in Scotland, and salmon farming is a major industry in Scotland. And the salmon are one of the benefits to us from salmon is that they accumulate omega-3 fatty acids, and we eat the salmon. But they accumulate it from algae, and they have to be fed food that contains omega-3 fatty acids. And there's a real issue of scooping up all the algae and sandy eels to feed the salmon to give them the omega-3 fatty acids. So Jonathan Napier and his colleagues at Rothamsted Research are engineering camelina, which is an oilseed crop, to make omega-3 fatty acids. So this involves them adding in several new genes to provide the enzymes to synthesize omega-3 fatty acids. And they're well on the way to producing camelina that will provide this as feed for the salmon farming industry. I think this is a very exciting potential, but of course in Scotland we've currently banned production of any GM crops, so at the moment this cannot be grown in Scotland. My last example is from the John Innes Centre again, And there they have a very ambitious program, which is to increase the nitrogen availability to crops. So as you can see from this diagram, map of the world, where in red shows the countries where nutrient is limited, nitrogen availability limits crop production. So it's very limited where it's red. In blue it's nutrient and irrigation limited, and green about 50% of crop yields are achieved because of nitrogen limitation. And Giles Oldroyd at the John Innes Centre is developing a program, and what they want to achieve is they want to engineer crops like wheat so that they can fix nitrogen by forming the nodules on roots that legumes like peas form, and they host nitrogen-fixing bacteria. So the idea is that they can understand the biology of production of nodules in the pea. They can then genetically alter crops like wheat and rice so that they can fix nitrogen as well. And that program has a lot of funding from, for example, the Gates Foundation. So there's a lot of interest from, in this case, I would say a beneficial NGO. I think NGOs have been getting a bad name, but the Gates Foundation is very interested in increasing the yields of crops to deal with the problem of the challenge of food production in a worldwide level. So the new breeding technologies using these CRISPRs, these molecular techniques, give us the tools which really they're available now to make simple gene mutations. So these mutations could exist naturally. So for example, they could exist in a poor quality breed of or cultivar of wheat, and you would want to bring that in to your high quality frontline breeding program, but it would take many years to move a single gene or a variation from one breed to another, but you could use this technology to introduce it directly into your breeding stock. You can move the variants between breeds, as I say, and if you move just a single mutation, you're not moving a whole bunch of genes along together, so it's much more efficient. You can move gene variants between species, and that can't be achieved by breeding. For example, Jonathan Jones at the John Innes Centre is interested in blight resistance in potatoes, and he's looking for blight resistance in distantly related wild plants so that he could introduce that gene from a wild plant that's resistant to blight into potato. You could also introduce novel genes that are not already in a crop, and that's the example of getting an oilseed crop to make omega-3 fatty acids. So they're completely novel pathways, but it's quite possible to do so. So I think we're at a point now where there's been a parallel increase in knowledge of genome sequences, gene structure and function in crops and farm animals, and also in their diseases, which is resulting in us being poised at the point now where we can use genome editing and genetic modification to increase productivity faster and in novel ways. But I don't think this means that what we've heard about already about hybrid wheat, for example, so on, that's going to go out. These are new technologies that complement the existing technologies. But what are the most useful and practical applications? We really need to think, how are we going to use these technologies? And we heard about rotating crops. We can't just suddenly introduce resistance to something, put it into all our breeds, and not expect the pathogens to overcome that resistance. We have to use them wisely. And how will genome-edited crops be regulated? This is really up in the air at the moment. In the US, the regulation of genetically modified crops is all based on the way the crops were genetically modified. And actually, genome editing doesn't really fall under that regulation. So it's quite possible that genome editing will be used and it won't need to be regulated. Now, I think we need to have some form of regulation because we need to consider things like herbicide tolerance and any risks that are associated with them. We can't just not regulate the use of new technologies. Then there's the whole question of acceptability to consumers. And we all know that GM has got a bad name, but I think that there is the opportunity now to revisit all that discussion with consumers. And on the whole, I find that many consumers are willing to consider using new technologies if they can see the benefits, and that we shouldn't listen too much to the lobby groups that are against any sorts of technologies. And will these more complex combinations of genome editing, which is very small genetic changes, and GM, will they become accepted? And I think if you want more information, these are a couple of people at the John Innes Institute in Rothamsted who would be very happy for people to contact them to find out more about the programs going on at those institutes. And I'd just like to apologize for having a tickle in my throat while giving the talk.